DNA backbone is made up of deoxyribose sugars and phosphate groups, which are covalently bonded to each other. The polynucleotide strands have directionality either from 5' to 3' or in the opposite direction, in which 5' and 3' refer to the carbon ring numbers. The phosphates attached to the 5' of the carbon ring are on the 5' end, and the phosphate attached to the 3' of the carbon ring is on the 3' end. Nitrogenous bases, which are covalently bonded to the 1' of the carbon ring, form hydrogen bonds with their respective pairs, adenine with thymine and guanine with cytosine. The purines, which are adenine and guanine, pair with the pyrimidines, thymine and cytosine. Guanine and cytosine form 3 hydrogen bonds with one another, while adenine and thymine only form 2. In eukaryotic cells, the DNA replication takes place inside the nucleus, while replication occurs in the cytoplasm for prokaryotic cells. To initiate the DNA replication, helicase unwinds the twisted DNA helix by breaking the hydrogen bonds between the nitrogenous bases. After the DNA is unwound, the single-stranded binding proteins bind to the unwinded portion of the DNA in order to prevent the strands from attaching together. As helicase unwinds, the DNA from behind, topoisomerase, replaced farther to the front of the DNA, prevents supercoiling by breaking and sealing the strands. Primase then creates primer, which is a small strand of RNA, and DNA polymerase builds new strands of DNA, starting from the primers in the 5' to 3' direction of the template DNA. The DNA polymerase 3 in charge of the DNA in 5' to 3' direction creates a leading strand, while the DNA polymerase 3 on the opposite side, with DNA from 3' to 5' direction, creates a lagging strand. As the newly synthesized DNA elongates, DNA polymerase 1 removes the primers. After all the DNA has been replicated, ligase connects the gaps between the DNA fragments. Mutations that can occur during DNA replication can be categorized into 3 groups: substitution or point mutations, insertions or deletions, and frameshifts. The first category, point mutations, occur when there is a substitution of one nucleotide in the DNA sequence. This can be further broken down into 3 more types of substitutions. In a silent mutation, a codon in the mRNA is changed, but it still codes for the same amino acids as intended, so there is no noticeable impact. In nonsense mutations, a codon is changed from its original codon into a stop codon, typically resulting in an unfinished protein. The final point mutation is called a missense mutation and occurs when a codon is swapped with another codon that codes for a completely different amino acid, which has varying results. Sometimes the protein has to be discarded due to the severity of the change. The second category of mutations is insertions or deletions. An insertion is an addition of genes to the DNA sequence which shifts the following sequences of nucleotides and results in different groupings of codons. Deletions have an opposite effect, and a codon is deleted from the DNA, which also results in different grouping of codons. The third category is a frameshift, which is actually a result of insertion or deletion mutations. A frameshift occurs when the insertion or deletion of codons results in a sequence in which the number of nucleotides is no longer divisible by 3, causing a shift in grouping. Like DNA, RNA has a backbone that consists of a sugar molecule that is bonded with a phosphate group. The sugar that is used in RNA is called a ribose sugar, which has a hydroxyl group attached to the 2' of the sugar. The 4 nitrogenous bases of RNA are adenine, guanine, cytosine, and uracil. Uracil takes the place of thymine during transcription. mRNA, the product of transcription, is synthesized in the 5' to 3' direction, using the 3' to 5' DNA strand as a template. During transcription, helicase is used to unwind DNA strands and initiate transcription. An RNA polymerase travels along the DNA and synthesizes mRNA that is complementary to the template strand. Transcription in eukaryotes occurs in the nucleus while it occurs in the cytoplasm for prokaryotes. In the process of transcription, RNA polymerase makes a complementary RNA strand based on the template DNA strand as helicase unwinds the DNA. This RNA strand is known as the messenger RNA or mRNA. In the RNA strand, introns, which are portions of RNA that do not code for genes, are excluded. After transcription of the mRNA is complete, the mRNA exits through the pores in the nuclear membrane and enters the cytoplasm. The mRNA is then attached to free ribosomes, which are made of our RNA. Translation occurs in the cytoplasm of both eukaryotic and prokaryotic cells. Since both transcription and translation occur in the cytoplasm in prokaryotes, prokaryotic transcription and translation can occur at the same time. During translation, tRNA molecules, each carrying an anticodon and an amino acid, attach to the complementary codon that lies on the mRNA. The amino acid that is attached to the top of the specific tRNA then forms a peptide bond with the other amino acids in the polypeptide chain. As the tRNA leaves, the amino acid that is previously attached to it is left behind in the chain. The ribosomes continue this process until a stop codon in the mRNA is reached, ending translation. The result of translation is a chain of amino acids which then undergo the folding process that results in the formation of a protein.